Today we are going to see about chromosomal aberration, introduction, types of chromosomal abnormalities, numerical chromosomal abnormalities, structural chromosomal abnormalities. Introduction. Chromosome abnormalities are changes resulting in a visible alteration of the chromosomes. Most chromosome aberrations are produced by mispair of broken chromosomes, by improper recombination, or by malsegregation of chromosomes during mitosis or meiosis. Types of chromosomal abnormalities. A chromosomal abnormality present in all cells of the body is called constitutional abnormality. If it is present in only certain cell or tissue, it is called somatic or acquired abnormality. Constitutional abnormalities are the result of an abnormal sperm or egg or maybe abnormal fertilization or an abnormal event in the early embryo. An individual with a somatic abnormality is a mosaic containing cells with two different chromosome constitutions, with both cell type deriving from the same zygote. Chromosomal abnormalities, whether constitutional or somatic, mostly fall into two categories: numerical abnormalities and structural abnormalities. Occasionally, abnormalities have been identified in which chromosomes have the correct number and structure, but represent unequal contributions from the two parents. Numerical chromosomal abnormalities. Three classes of numerical chromosomal abnormalities can be distinguished. They are polyploidy, aneuploidy, and mixoploidy. Polyploidy. In human pregnancies, about 3% are triploids, the most usual cause in two sperm fertilizing a single egg. Sometimes the cause is a diploid gamete. Triploids are seldom survived to term and the condition is not compatible with life. Tetraploidy is much rarer and always lethal. It is usually due to failure to complete the first zygotic division. The DNA has replicated to give a content of 4 carbon, but cell division has not taken place as normal. Although constitutional polyploidy is rare and lethal, all normal people have some polyploidy cell. About two-thirds of human triploids arise by fertilization of a single egg by two sperm. Other causes are a diploid egg or sperm. Most human triploids abort spontaneously. Very rarely they survive to term but not beyond. Aneuploidy. Euploidy means having complete chromosome sets. Aneuploidy is the opposite, that is, one or more individual chromosomes extra or missing from a euploid set. Trisomy means having 3 copies of a particular chromosome in a otherwise diploid cell, for example, trisomy 21. In Down syndrome, monosomy is the corresponding lack of a chromosome. For example, monosomy X in Turner syndrome. Cancer cells often show extreme aneuploidy with multiple chromosomal abnormalities. Aneuploid cells arise through two main mechanisms. Non-disjunction. Failure of paired chromosomes to separate in anaphase of meiosis I, or failure of sister chromatids to disjoin at either meiosis II or at mitosis. Non-disjunction in meiosis produce gametes with 22 or 24 chromosomes, which after fertilization by a normal gamete make a trisomic or monosomic zygote. Non-disjunction in mitosis produce a mosaic. Anaphase lag. Failure of a chromosome or chromatid to be incorporated into one of the daughter nuclei following cell division. As a result of delayed movement, lagging during anaphase, chromosomes that do not enter a daughter cell nucleus are lost. Mixoploidy. Mixoploidy includes mosaics, an individual possesses two or more genetically different lines derived from single zygote, and chimerism, an individual has two or more genetically different lines originating from different zygote. Abnormalities that would be lethal in constitutional form may be compatible with life in mosaics. Mosaics have two or more genetically different cell lines derived from a single zygote. The genetic change indicated may be a gene mutation, a numerical or structural chromosomal change, or in a special case of lionization, X inactivation. Aneuploidy mosaics are common. For example, mosaicism resulting in a proportion of normal cells and a proportion of aneuploid cells can be ascribed to non-disjunction or chromosome lag occurring in one of the mitotic division of the early embryo. Polyploid mosaics, example human diploid or triploid mosaics, are occasionally found as gain or loss of a haploid set of chromosomes by mitotic non-disjunction is most unlikely. Human diploid or triploid mosaics most probably arise by fusion of a second polar body with one of the cleavage nuclei of a normal diploid zygote. Having the wrong number of sex chromosomes have far fewer ill effects than having the wrong number of any autosome. 47XXX and 47XYY. People often function within the normal range. 47XXY men have relatively minor problems compared to people with any autosomal trisomy and even monosomy in 45X. Women has remarkably few major consequences. In fact, since normal people can have either one or two X chromosomes and either no or one Y, there must be special mechanisms that allow normal function with variable numbers of sex chromosomes. In the case of the Y chromosomes, that is to because it carries very few genes whose only important function is to determine male sex. For the X chromosome, the special mechanism of lionization controls the level of X encoded gene products inside independently of the number of X chromosomes present in the cell. Structural chromosomal abnormalities. Chromosome breaks occur either as a result of damage to DNA by radiation or chemicals, for example, or as a part of mechanism of recombination. In G2 phase of the cell cycle, chromosomes consist of two chromatids. Breaks occurring at this stage are manifest as chromatids breaks affecting only one of two sister chromatids. Breaks occurring in G1 phase, if not repaired before S phase, appear later as chromosome breaks affecting both chromatids. Cell have enzyme system that recognize and if possible repair broken chromosome ends. Repair can be either by joining two broken ends together or by capping a broken end with a telomer. Cell cycle checkpoint mechanism normally prevents cell with repair chromosome break from entering mitosis. If the damage cannot be repaired, the cell commits suicide. Chromosomal abnormality due to uniparental chromosomes. The rare abnormalities described below demonstrate that it is not enough to have the correct number and structure of chromosomes. They must have the correct parental origin. 46XX conceptuses in which both genomes originate from the same parent never develop correctly. For some individual chromosomes have both homologs derived from the same parent also cause abnormality. A small number of genes are imprinted with their parent origin and are expressed differently accordingly. Uniparent disomy UPD affecting a single pair of homologs goes undiagnosed if the result is not abnormal but is detected for chromosomes for which it produces characteristic syndromes. UPD can be isodisomy where both homologs are identical or heterodisomy where they are derived from both homologs in one parent. The usual cause is thought to be trisomy recessive, a concept that is trisomic and would otherwise die. Occasionally loses one chromosome by mitotic non-junction or anaphase lag from a totipotent cell. The euploid progeny of this cell form the embryo while all the aneuploid cells die. If each of the three copies has an equal chance of being lost, there will be a 2 in 3 chance of a single chromosome loss leading to the normal chromosome constitution and 1 in 3 chance of uniparental disomy. Uniparental disomy may possibly arise by selection pressure on a monosomic embryo to achieve euploidy by selective duplication of the monosomic chromosome. Thank you.